Hello and welcome to our video explaining the process of meiosis. The cell starts in interphase. As you can see, there are 3 homologous chromosomes in the nucleus. These chromosomes are in G1. They have not gone through synthesis yet, as the DNA has not duplicated. The beads represent genes on these chromosomes. These genes code for various proteins which create physical traits. Some genes look similar and code for the same category of physical trait, but have different alleles. However, every homologous chromosome in a homologous pair will have the same alleles as the other, as the DNA was replicated exactly. The chromosomes go through synthesis, in which the DNA is duplicated. Now you can see 3 homologous pairs of chromosomes. Meiosis 1 starts with prophase 1, when the chromatids are lined up in homologous pairs and cross over each other. When this happens, genes can be exchanged between the chromatids to create variety. The genes that are closer to the centromere are less likely to be exchanged, and the ones that are further are more likely to be exchanged. This has helped with mapping the genome, as we can tell which genes are in what order in relation to the centromere due to how often they are exchanged. During metaphase 1, the chromosomes line up in pairs in the center. The order of these pairs can be different in order to create an independent assortment. In other words, the chromosomes line up independently of each other. For example, here's another possible way that the chromosomes could have lined up. In anaphase 1, these chromosomes are pulled apart by spindle fibers and 2 nuclear membranes are formed. We will now just focus on one of these new cells, but remember that the same process of meiosis 2 is happening in both cells. We will come back to the other cell later.
Congratulations, you've made it through meiosis 1. Now you're on to meiosis 2, the second division that separates the sister chromatids. But keep in mind, these chromatids may not be identical due to crossing over in prophase 1. In prophase 2, the chromosomes condense, the nuclear membrane dissolves, and the centrosomes move to opposite poles, which is perpendicular to the first time in prophase 1. Then, in metaphase 2, the spindle fibers from opposing centrosomes attach to the chromosomes at the centromere and align them along the cell equator. In anaphase 2, the spindle fibers contract and separate the sister chromatids, and the chromatids, which are now called chromosomes, move to opposite poles. And then, in telophase 2, the chromosomes decondense, the nuclear membrane forms, and the cells divide, or otherwise known as cytokinesis, to form 4 haploid daughter cells. The final outcome of meiosis is the production of 4 haploid daughter cells. These cells may all be genetically distinct if crossing over occurs in prophase 1, and that causes the recombination of sister chromatids. Bye!